Hi, my name is Nureen Yazina binti Mama Fauzi. My metric number is F1880282. My research findings is about the dietary effect of the mulberry pellet on the hematological analysis and also the biochemical analysis in rabbits. First, we start with the research background. Mulberry tree is widely distributed all over regions and also suitable in the tropical and also into the temperate conditions. The production of the rabbits is cost effective. It is due to the rapid growth of the rabbits, the short period gestations, and also the low cost of production in rabbits. Some research suggested that the high quality protein in the mulberry could be used into the feeding livestock. It is because mulberry is cheap in sources of minerals, fiber, and also protein. A rabbit diet is essential to its overall health issues. Next, we move to the part of objective of this experiment. The first objective is to evaluate the effect of mulberry pellet on the hematological and also the mulberry pellet on the biochemical analysis in rabbits. Problem statement of this experiment was rabbit production is not widely commercialized, but it is highly profitable and it is good for the human consumption. The major problem that faced by the rabbit farmers, are the high cost of the commercial pellet. Incorrect feed nutrition will leading to the suffering from the health problems in the rabbits. Mulberry pellet will be tested for the effectiveness in improving rabbit blood profile that resulted in producing more natural product that is satisfying by improving the health of the rabbits. The hypothesis of this finding is there are significant difference and there is no significant difference in the mulberry pellet feeding of blood profile consistence in rabbits. Literature review. The Morus alba is the scientific name of the mulberry. It is a dominant species in the Moraceae family that found in a wide range of climate from the tropical into the temperate conditions. Rabbits, or the scientific name is the Oryctolagus cuniculus, has one of the lowest fat and highest protein amount consumed meat for humans. In the mulberry diet high in fiber content, it is suitable for the rabbit for the gastrointestinal health. Monogastric, ruminant, and also rabbits eat protein-rich forages. Methodology of these experiments. This experiment is classified into the three dietary treatments which are control group, treatment 1 group, and also treatment 2 group. The control group will be given 100% of the commercial pellets. T1 or treatment 1 group will be given with the combination of the mulberry pellet and also the commercial pellets, while the last one is the T2 group will be given with the 100% of the mulberry pellet formulations. In about the sample preparation, the first is fresh mulberry leaf and stem will be dry, blend, and also grind into the powder form. The mulberry formulation will contain 70% of the dry leaf and also 30% of the dry stems. It was mixed with other ingredients such as molasses, 5% of tapioca starch, and also 1% of the sodium bicarbonate. Then the mulberry formulation will be pelletized using the pelletizing machine with the diameter 0.4 millimeters. The experimental designs. 9 to 10 days old New Zealand white rabbits will be put in the individual cage. This feeding trial will be conducted within 16 months or 2 months. Pre-feeding trial and post-feeding trial blood collection conducted by following their weight. It is by calculate using the mean blood volume and also the body weight of the rabbits. It is because only 0.01% of the total blood volume may be collected from the rabbit within 2 weeks. Next, the blood collection will be conducted into the two analyses, which is the first one is the hematological analysis, will be tested by using the machine MyPick CBC test, and the second one is the biochemical analysis will be analyzed using the IDEXX Biochemical Test Machines. Next, about the data analysis. Data analysis will be used with the SPSS, 1B ANOVA, Turkey, and also Duncan test. Next, we move to the results and also the discussion parts. According to the figure 4.1a, T1 recorded the white blood cell and lymphocyte were average values than the control and also the T2 groups. Monocyte in the control group and T2 group were average compared to the T1 groups. Granulocyte in the T1 with 2.75 was in the normal range other than in the C and also the T2 group. Lymphocyte percent, also the granulocyte percent in the control group was recorded the typical values range with the average 42.2 and 37.69 respectively. Monocyte percent levels in the T2 was better than the C control and the T1 treatment 1 with 20.5 and 16.25 respectively. However, there is not in the normal range. Next, we move to the part of the according to the figure 4.1b. Red blood cell in the control group and also the treatment 1 group were average compared to the T2 group, which is the treatment 2. HGB value in the control and also in the T1 were recorded to the nearest to the value average value. HCP in the T1 recorded the average range value than the C and also the T2. MCV in the control group with the recorded state the values 89.7 was in the normal range than T1 and also T2. MCH in the control group with 18.4 was recorded the nearest average values compared within the two group T1 also T2 group. Next, we move to the figure 4.1c. MCHC value in T2 with 20.35 was recorded to the nearest average values compared to the control group and also T1 group. RDW and PLT in T1 and also T2 were recorded in the average values than the control group. MPV and PCP in control and T1 group was displayed as the nearest average values. PDW in T1 was recorded in the normal average. However, T2 was displayed the value in the normal range. This findings show that the T1 was the optimal formulation for the hematological analysis than the others group, which is control and also T2 group. Alright, next we move to the part of the biochemical analysis results. According to the figure 4.2, bone or blood urea nitrogen in T1 and T2 were recorded with the nearest average value compared to the control group, that being the highest one. Glucose or GLU and ALT values in the T2 were displayed the average value range compared to the other groups C and also T1. TP or total protein in T1 recorded the average values vary with the nearest values. This findings may shows that the T2 was the optimal formulation for the biochemical analysis within than the other groups C and T1 groups. Conclusion of this research indicates that a new type of rabbit pellet has excellent potential and suitable for the feed diet that improve the parameters level of blood constituents. In hematological and with the tremendous potential formulation, T1 is the most excellent than the control and T2 group. However, T2 state the superior in biochemical analysis. The cost of producing this pellet was significantly lower than the other commercial pellet and is much easier to obtain. The recommendation of these findings. To ensure that the specific composition of the mulberry pellet is maintained off the UMK Jeli campus, mulberry plants should be planted and treated with a fertilizer or any other nourishment nutrient. The next is mulberry trees need to be replanted regularly, maintain their maximal fruit productions, maximum leaf density, and overall health at their highest levels. The last one is for the sample to run smoothly. The laboratory apparatus and machine must be regularly operated and updated, such as grinder machines, pelletizer machine, and also chopper machines. This is the reference we use in the findings. Thank you.